This Neurobinar of April. I am Ingrid Miucci, the Communication Manager of Neuromarseille, and I am delighted to welcome you here. Before we begin, here is a quick reminder of the Neurobinar program. This is a 1-hour conference in 2 steps. So before we will begin with 10 minutes of Live My Life, where 2 PhD students present the author's work. This will be followed by 5 minutes of question, and then there will be a presentation for 20 minutes with a researcher, followed by 15 minutes of questions. For this edition, we are delighted to welcome Benoît Drouillas and Nicolas Hugues for the PhD swap. So Nicolas Hugues will begin by introducing the work of Benoît Drouillas from INT. Actually, Benoît Drouillas' thesis is on differential contribution of NAV 1.1 and NAV 1.6 channels to bistable properties within the lumbar spinal locomotor network. He is supervised by Frédéric Broca. And then Benoît Drouillas will present Nicolas Hugues' thesis, who is working on impact of various endurance exercise regimens and effects of their combination with cognitive training on neuroplasticity after cerebral ischemia. He is supervised by Professor Eric Berton and Jérôme Laurin from INMED. After this, Jean-Christophe Roux from MMG will present his research on optimization of gene therapy and Rett Syndrome. Nicolas, it's up to you to start.
So, hi everyone. So I'm going to introduce Benoît's works. So Benoît works at the Institut de Neurosciences de la Thyroïde, where he investigates the locomotion on mice. He is interested in the lumbar part of the spinal cord, which support 2 main functions essential for locomotion. So the first is the Motorism Genesis, which is coming from a group of ventral interneurons located in the L1, L2 segment, which form CPG, which are central pattern generators. Once produced, the rhythm is spread among the spinal cord to recruit motoneurons, which support the second function, which is the production of the postural tone. These 2 functions are supported by 2 distinct properties. So the CPG cells, which express an autorhythmic activity called pacemaker property, and the motoneurons, which express a self-sustained firing called plateau property. Their particularity is that they both rely on the same current, the persistent sodium current called INAP, which, when blocked, as we can see on the right, can lead to the abolition of both properties. So the aim of Benoît's works is to determine which sodium channel underlies the persistent sodium current INAP. The literature learns them that NAV 1.6 is the main sodium channel expressed in motoneurons, as we can see 100% of motoneurons express NAV 1.6, and 80% of them also express NAV 1.1. Moreover, other studies show that mice lacking NAV 1.6 suffered from locomotor and postural deficits, as we can see right there. Hence, regarding the literature, Benoît's hypothesis is that NAV 1.6 modulates the bistable property, the plateau, and the pacemaker properties, which are both essential. To verify this hypothesis, he used a patch clamp recording on both CPG interneurons as well as motoneurons on spinal cord slices in different conditions. So first, he used NAV 1.6 knockout mice. Then he used a pharmacological approach with NAV 1.6 specific blocker. And then finally, he used gene therapy in order to delete specifically the NAV 1.6 from the motoneurons. In the first result, Benoît found that in wild type animals, 80% of motoneurons express a plateau, while in a NAV 1.6 knockout, only 20% of motoneurons express a plateau. Surprisingly, on the other side, the proportion of pacemaker cells is not impacted by NAV 1.6 knockout. So this data indicate that NAV 1.6 is a key channel in motoneurons, but might not play a role in pacemaker cells. To exclude the possibility of a compensation mechanism, Benoît switched to a pharmacological approach. When a specific blocker of NAV 1.6 was applied on wild-type animals, Benoît found, again, the same result with a huge reduction of motoneurons expressing a plateau, as we can see right there, while no difference in cells. So we can conclude that NAV 1.6 is essential for the plateau property of motoneurons, but not for pacemaker cells. Finally, Benoît tried to be more specific as possible by the means of AAV injection coupled with short mRNA targeting NAV 1.6 specifically in motoneurons, as we saw previously. And thanks to a fluorescent reporter, he was able to record only the infected motoneurons. And in this context, the proportion of motoneurons expressing a plateau property is drastically reduced, as we can see here. One more time. In addition to the previous results, this one confirms what was found previously. So to summarize, motoneurons express the plateau property, which is related to the post-Ralton production, and this property relies on NAV 1.6 channels. But in contrast, the pacemaker property related to the Motorism Genesis, which is expressed in interneurons of CPG, doesn't rely on this channel. So, So the next step of his work will be to determine if NAV 1.1, which is present in 90% of motoneuron, which is expressed in 80% of motoneuron, play a role or not in the motoneurons, the interneurons. Sorry. Okay. Thank you.
Okay, thank you, Nicolas. Now let's go for 5 minutes about your project. So on your side, you are working on the effect of endurance training intensity on neuroplasticity, as well as on sensory motor and cognitive deficit induced by cerebral ischemia. Cerebral ischemia is the second cause of death in the world, and almost 17% of the model population has already suffered from a stroke. And this stroke induces both cognitive deficits and motor deficits in humans. While multiple cognitive training strategies are employed in clinics, their gains cannot always be transferred or maintained over the long term. However, recent studies show that physical activity has a high recovery potential by stimulating synaptic plasticity, neurogenesis, and angiogenesis in humans. That leads us to Nicolas' question, which is, can intense physical exercise reverse motor and cognitive deficits after a stroke? To answer this question, Nicolas is using a surgical approach to induce the cerebral ischemia in rodents. To do so, he's inserting a really tiny monofilament to occlude the middle cerebral artery, and he did it for 2 hours. By this way, he can deprive a part of the right hemisphere from oxygen and nutrients. This 2 hours duration has been set in order to reproduce the deficits you can observe in human. And here on the brain slice, you can see it shows us how big is the infarct, and this infarct affects both our hippocampus, striatum, and the cortex. With his model, Nicolas aimed to compare various training intensities to determine whether the actual clinical recommendation could be optimized or not. And to do so, he used an incremental test to determine what is the speed at the lactate threshold, because the lactate threshold gives us the transition between what is a moderate intensity activity and a high intensity activity. And by combining this parameter with the maximum speed, he is allowed to individualize the training intensity for each animal in accordance to their capacities and their weight. And by this way, it can set an individual parameters to dissociate moderate intensity training from high intensity training. Then he studied the muscle-brain crosstalk, but didn't observe any effect of the training on the hypsilisonate side. That could be explained because there are gliogenesis and angiogenesis, which are indicators of neuroinflammation, and which could block the neuroplasticity processes. Nevertheless, when focusing on the contrarresonance side, the seems to be more efficient. Indeed, what he first observed is the change in the microglia phenotype, which switched to the M2 phenotype, as you can see in the green dot here. And the M2 phenotype is well known to be implicated in functional recovery. Additionally, what Nicolas found is an increase in the FNDC5 proteins, which is a protein released by active muscles and which stimulates the TrkB BDNF neurotrophic pathway. At the same time, the phosphorylated form of the TrkB receptor, which is also a major actor of this cascade, is also increased. So together, such increases could contribute to beneficial outcomes due to their role in synaptic plasticity and neurogenesis. But the real question is, are these changes enough to reduce stroke-induced deficits? The answer is yes and no. Yes, because first, what is observed is a huge increase of the endurance performance characterized by an increase in both the speed at the lactate threshold here and the maximum speed after only 2 weeks of HIIT training. Moreover, this HIIT training allows a full recovery of the forearm grip strength. And here the red line shows you the complete recovery after 2 weeks of the stroke model animal after the HIIT. On the other hand, Nicolas performed cognitive task as the Nobel of General Cognition or the Y maze, but he didn't observe an improvement thanks to the HIIT. So even if the HIIT, as I told you, promotes contralesional neurogenesis, maybe the training period he used was not sufficient enough to reverse stroke-induced deficits. That's why his new objective is to combine this HIIT training with a daily cognitive training. And this kind of protocol has been previously showed to promote neurogenesis in safe animal. And his whole aim will be to determine whether a cognitive rehabilitation could potentiate its effect on neuroplasticity and redirect this effect on cognitive performance. Thank you for your attention.
Thanks a lot, guys. So now we are going to pass to the question and answer. So do not hesitate to raise your hand or to ask your question directly in the Q&A section. So in your Zoom interface, you have Q&A with like 2 bubbles.
Yeah.
There was a question from Lydia.
Yes, I thank you for your talks. I have a question on the thesis of Nicolas. So I was wondering, where have you examined the microglial reactivity in the hippocampus, striatum, cortex? So I missed this point. And have you looked at oligodendrocytes also, oligodendrogenesis?
So first, what is interesting is that muscle is able to produce such molecules as FNDC5, as Benoît said, which are able to go through the brain and to stimulate the BDNF TrkB pathway, which is known to be linked to neuroplasticity, and this neuroplasticity could be therefore implicated in cognitive performance. So it's only one pathway, but there are various and numerous that can play an important role and stimulate BDNF pathway, but also angiogenesis through VEGF or EGF1, for example. but in a second time, no, we actually didn't measured oligodendrocyte yet.
Thank you.
Okay, another question about the heat. Are there similar results in humans? Tu veux pas répondre?
Yes, in terms of endurance performance, yes, as we could see this previously. Only 2 weeks of high intensity interval training was enough to induce gains in physiological endurance parameters, such as the speed at lactate threshold or the maximal speed. So we can imagine that a longer period of training might permit to enhance on a longer time these parameters, maybe contrary to moderate intensity continuous training, for which a plateau phase could be observed after a few weeks.
Okay, another question from Geneviève. How do you differentiate the M2 from the N1 phenotype in microglia?
We can observe IBA1 protein and the phenotype change from kind of a star microglia to a more round phenotype, which permit to distinguish one from another phenotype of microglia.
Okay, and then what are the proof of a relation between neurogenesis and cognitive functions?
Actually, it's difficult to link the two. We are trying to make this link, as we can see on the last diapositive. Cognitive training can induce neurogenesis, but we want to see now if neurogenesis induced by cognitive training and exercise training can have an impact on the cognitive function. So through the Barnes maze, novel object recognition, or the Y maze.
Okay, another question. Is the time when running is performed important? Immediately after the MCAO or delay?
Yes, of course, it's really important. It has been shown previously that too early training could have deleterious effects on the brain. So it is important to wait a little bit. So here on animals, it's important to wait 1 day so we can begin the training, the incremental test at D1. And this is this window, this plasticity window, which is really important. It's to work early as possible, but not too early in order to have the best effect, but not to have deleterious effects. So not too early.
Okay. And again,
maybe a question to Benoît. There was the last one from Anne. If
Neurobinar is involved in postural tonus, is it linked to motor disorder like spasticity phenomenon? It's an excellent question, and you are totally right. A study published by the team in 2016 demonstrated the importance of neurobinar in the development of this of spasticity after spinal cord injury. So you're right. Neurobinar is really important in troubles as a spinal cord injury, and also epilepsy. Yes. Another question for you. The TrkB BDNF pathway, did you observe an increase of the receptor or the BDNF directly?
Yeah, we observed an increase of the receptor, so TrkB, and we thanks to Western blotting, and then we tried to measure BDNF through Western blot, but it was not possible. So we did this thanks to ELISA kit, and we observed an increase of brain-derived neurotrophic factors only in the contralateral cortex, in the ipsilegional cortex, sorry, in the right hemisphere for MC-AO group and for HIT groups. So maybe it's a spontaneous increase of brain-derived neurotrophic factors and not an increase thanks to training.
Now, what about the method?
Elisa,
okay.
Yeah.
Okay, thank you. We're going to stop here for the questions for the PhD students, but do not hesitate to ask your questions. They can respond to you. And we are going to pass to Jean-Christophe Roux, which will be presented by Laurent Fasano.
Okay, so it's a great pleasure for me to present Jean-Christophe Roux from the Marseille Medical Genetic Laboratory, located on the Timon campus. So Jean-Christophe works on the Rett Syndrome, which is a devastating genetic disorder leading to autonomic and cognitive deficits. Rett Syndrome is a rare genetic neurological disorder caused by mutation on the gene MECP2 located on the X chromosome. As a consequence, this syndrome occurs almost exclusively in girls. So to identify the mechanism underpinning this syndrome, Jean-Christophe group is studying mouse model of MECP2 dysfunction. And I want to say that this approach has been very successful and has led to the implementation of two clinical trials, phase 2, with a Rett patient. I would also like to point out that Jean-Christophe is very active in the scientific community and deeply involved in scientific communication. So I think that all this makes Jean-Christophe the perfect speaker for our webinar of today. So usually we say the stage is yours, but now we say the screen is yours. Go ahead.
Hi, everyone. Thank you very much for this very nice introduction, Laurent. And now I will talk about gene therapy, ultrasound, and Rett syndrome, of course. And as Laurent mentioned, I am working in the Marseille Medical Genetics Laboratory and in the team of the Human Neurogenetics headed by Laurent Villard. We have just to complete, we have three main topic projects in our team. The first one is early infantile epileptic encephalopathies. There is also another one, this is intellectual disability from genetic origin, and Rett syndrome. Yes, it's okay. Rett syndrome, as Laurent noted, leads to intellectual disability associated to troubles in locomotion, locomotor troubles, breathing dysfunctions, and this is one of the main problems for the patients because some of them, 20–25%, they die from breathing arrhythmia. In almost all cases, this is due to a mutation in methyl-CpG binding protein 2, MECP2. This is a monogenic pathology mainly. And as Laurent noted, only girls are affected because boys are more severely impacted by the mutation, and they die very early. They don't present, of course, obviously, as the phenotype, the Rett syndrome phenotype. Currently, there is no cure for Rett syndrome, and our team is working since many years, several years, on pharmacological approaches. And just for a short description of the syndrome, at birth, the baby is totally normal. Everything is fine until to 6 to 18 months. And they start if the baby is able to walk, they stop, to walk. If the baby is speaking, they stop to speak. And many different troubles start to arrive: epilepsy, breathing dysfunction, breathing abnormalities. And finally, we say it's apparent, there is an apparent stability later on, but it's a very handicapped pathology. But one interesting point has been, one paper has been published in 2007 showing that Rett syndrome, at least in mouse, a mouse model, is totally reversible. And you can see on the left part, there is a video here. This is MCP2 has been silenced using a lock-stop cassette. And the mouse is very sick. And if you don't do anything, after a few weeks, this mouse will die. And I wait a little bit, but in the left part, now, in the right part, this is exactly the same mouse. The scientist, the Scottish scientist, they have injected tamoxifen, and the cassette was released, and MCP2 was able to be activated even very, very late, close to the death. And if the mouse is totally normal, and it's impossible to distinguish this mouse to a wild type 1 now. It means that if 100% of cells express MCP2 even late during the pathology, the syndrome is reversible in mouse, in a mouse model, to be precise. This is why we started a project of using AAV9 vectors. And this project has been funded by the French Téléthon, AFM French Téléthon, and we are using AAV9 vectors with MCP2. I will call MCO because this is a codon optimized version of MCP2. And we are using two mouse models. The first one, this is a male, so knockout one, very severe model. And the second one, this is a female models. The real genetic model, this is heterozygous female models. And since we are interested by translational research, we're interested to treat the mice only when they start to present a phenotype. We waited till the mouse start to exhibit the first sign of the pathology. In male, it's around 30 days, it's quite close. In females, this is after several months. Then we treated the mice with the knockout male mice with the AAV vectors, and what you can, as you can see here, yes, on the top, here this is a body weight after treatment. In the untreated mice, you see the body weight is going down and down, and the mice are dying. This is a knockout one. If you look in the upper part, this is a wild type mice. So they are growing, this is normal. And the treated one, you can see here, they maintain the body weight quite a long time, and the body weight is increased and maintained. If you look at the survival here, this is untreated knockout mice. Here you have the wild type mice, and here you have the treated one with the AAV9 vectors. It works, survival is improved. And if you look down, this is the dark, the black square. You can see the knockout mice progressively when they grow. They increase, the number of apneas are increasing drastically. And if you look at the treated one in white, it's totally reduced, and there are almost no more apneas. This is good, but the problem, as I explained before, we try to reach the 100% of cell infected in the brain. And here, this is the pictures. You have the knockout mice, you have the wild type mice, and you can see that even in the AAV9 treated knockout mice, this little dot, this is MCP2. And obviously, if we counted, you can see that only 8 to 12% of neurons are infected. It means that this is still a sort of miracle because only few cells, only 8 to 12% are infected, and we got such a good improvement. Now we did the same with a female, the heterozygous female mice, and this is a real genetic model. And here you can see that for apneas, for instance, after several months, several weeks, you can see that the number of apneas in gray, sorry for the color code, but this is from the paper, but in gray, you can see that the number is increasing progressively in untreated mice, but in black, you can see that after several weeks, the number of apneas is quite stable and pretty close to the wild type one. Down, you can see this is locomotion as an example. On gray, at the gray part, you can see locomotion hour per hour during the day and during the night. And if you look at the locomotion in the untreated mice, the heterozygous mice, you can see that the locomotion is reduced. The distance traveled is really reduced compared to the wild type, and the treated one, they travel much more than the untreated one. It works, it's fine, but again, what we found in this case, I could explain, but it's a little bit complicated. But we found that the treatment, the AV treatment in heterozygous females induce inflammation and induce apoptosis at a special dose. When we start to reach efficacy, strong efficacy in vivo, we got also a liver inflammation and apoptosis, which is not good. Then altogether, coming from the knockout male mouse, there is a need to improve there is a need to improve the number of infected cells, and also for the females, coming from the female study, we have to work to decrease the liver toxicity. How it's possible to do this, to increase efficacy of infection in the brain? Well, obviously, we could directly inject the virus in the brain. This can be done, but and it works. It's pretty nice. But we believe that this protocol is complex and can only be used in lethal pathology, and it will be difficult in Rett syndrome to use this process, except that if we could prove that our vector is able to make a sort of miracle. Then it's necessary now to help the vector to cross the BBB, the blood-brain barrier, easily. And this is what you can see here, the blood-brain barrier that protects the brain from virus, bacteria, and big molecules. And this is necessary. But we decide to collaborate with physicists. This is 3 colleagues. One is located in Marseille, Laboratoire of Mécanique Acoustique, and 2 are located in Saclay, Paris-Saclay. And because they developed a technology, they are improving a technology using focused ultrasounds. The idea is basic. The basic idea is to inject in the blood, in the systemic injection of microbubbles, we'll come back later, and to provide with a special transducer, focus ultrasounds, and the focus ultrasounds can check and create a sort of cavitation with the bubbles, and the bubble progressively will push this blood-brain barrier to open progressively the blood-brain barrier to increase the penetration by the big molecules, and in our case, by the AAV vectors. As an example, what is interesting with this technology, depending on the acoustic pressure that you provide, you can go, if you have very low intensity, you can have a sort of neuromodulation, and people are using this technology to stimulate several brain areas. If you use too much, a huge acoustic pressure, you can burn and destroy the tissue like a gamma knife. It's another way, but you can burn and destroy a tissue. It can be interesting also for a medical doctor, but in our case, we are in between, and the idea is to enhance drug delivery. Just again, to show you the cavitation creates this kind of, I mean, the bubbles has to push the blood-brain barrier and to open progressively. And we have obviously to avoid that the bubbles explode, because any kind of explosion will create bleeding hemorrhage. Then this is our setup in our animal department. You can see here, this is an automated system, and we can select the brain area that we are interested in, or select, in our case, we have to treat the whole brain. This is, we are working this way, but it's also possible to use this technology just to focus in a special part of the brain. Here, this is the microbubbles that we are using, the SF6. And here, this is just, at the right part, this is just an example. I mean, this is where the focus ultrasound are just, the ultrasounds are focusing in the brain. And the idea is to move and to instead of to target the whole brain. Another important, really important point, this is the blood-brain barrier naturally is very important and has to be open for a short time. And depending also of the acoustic pressure that we could use, you can open the blood-brain barrier for several days, which is not good. And in our case, we are more interested to open the blood-brain barrier for several hours instead of to increase the AAV, the viral infection. Then we started, and at the beginning, several years ago, we started by using Evans blue. This is a coloration, natural coloration, not able to cross the blood-brain barrier. And if you inject this, this colorant in the blood, in circulation, the brain is totally white and you don't see anything except if the blood-brain barrier is affected. And we used initially several level of acoustic pressure from 0.45, 0.65. And here you can see this is where we target the brain with the ultrasounds. It's pretty blue, and it was too much. This is, to be honest, because 1 megapascal, we got bleeding and we killed several mice, but 0.65 was enough to get blue, to open the blood-brain barrier, and also to maintain the mice living for several days without any problem. Down, you can see this is AAV, this is wild-type brain from wild-type mice, and the little dot that you can see here, this is the GFP. This is AV9, CMV GFP. And here, the little dot, this is where the expression was observed in the brain. Here, this is exactly, not exactly, this is another brain, another mouse that has been injected, but we use such acoustic pressure and the microbubbles too. And you can see this is quite impressive. The number of infected cells, the increase that you can see on the pictures. We did it again, and we start to count the number of infected cells with or without focus ultrasound. And here, that was the first counting that we did, and we had at this time close to 2.3 to 3 times the number of infected cells in the prefrontal cortex, striatum, in the motor cortex, hippocampus. But now, with our last studies, we are close to 4 times to 5 times, which is even better. This is very good. We also check for if there are some side effects, and we haven't seen no brain inflammation using hematoxylin eosin, no apoptosis. We haven't seen any sign of apoptosis anywhere. We didn't see any activated microglia, no bleeding, which is good. Then it seems to be pretty safe. This is 2 days after injection of the virus, microbubble, and AAV and focus ultrasound stimulation. Now we came back to our therapeutic vector, what I presented before and with the knockout male mice. And we injected now the AAV9 vectors to these mice. Here, this is mice, they received just the bubbles. They received the PBS and received the ultrasound. And you can see the survival. And now, if you remember from the picture that I presented, the median of survival without any focus ultrasound was close to 90. And now we are at 127 days, which is much more, this is a pretty strong increase. And now we are looking for behavior to see if all the parameters that we measured previously, like breathing, locomotion, circadian activity, grip strength, etc., all these parameters should be, if everything is logic, should be improved compared to what we got without any focus ultrasound. And to show that it's not just something that you can use for fun, to improve our results in animals, in rodents, there is this kind of apparatus in USA, and this is the same, not exactly the same, it's much bigger, but this has been built for human patients, and there is an MRI to guide exactly where in the brain you want to apply to focus the ultrasounds. And there are right now, currently 35 clinical trials ongoing with this kind of machine setup. And mainly this is for neurostimulation or for burning different parts of the brain, but there are several clinical trials ongoing with the goal of to opening the blood-brain barrier. And one of our collaborators is also working on the development of this kind of machine for monkey, for primates. And this could be also, will be interesting for us if we want to translate our results from rodents to patients. And just to finish, just to show you, this is just, I have to thank all the people in our team working in this, the one that are working now and the past one, and our collaborators, and also the funding that we got, especially one from the region, the PACA region, that with this funding, we were able to buy the setup and to implement the setup in our animal department. Thank you very much for your attention. And now, if you have any question, I will be happy to answer if I can.
You already have 3 questions for you. The first one is for Benoît. You see it? It's okay?
Yeah.
You can just read it for
Benoît? For Benoît, or I start directly?
No, you start the second one, naive question.
Yeah. Can we detect this syndrome with an amniocentesis? Yes, of course. This is a big question. Yes, there is a big issue. There is a paper now published several days ago in Nature's. I mean, people try to, they are thinking that if we are able, which is easy, by genetic process, you can detect the mutation, it's quite easy. But the problem, amniocentesis, you can have abortion by doing this, it would be difficult first, even if technically it's possible. I don't believe this will be used in human being, and it would be difficult to justify that to do it only for Rett syndrome and to not do it for other terrible pathologies. This is why, yes, it's possible to detect, but nobody are doing this. I'm not sure that it will be fine to do it in the future. Do you try other AAV? No, because there is a PHPB that, I mean, the people ask if I used other AAV subtype. I know that there are papers showing that the PHPB, this is another vector, even better to cross the blood-brain barrier naturally. But there are one publication in Rett syndrome, this vector in this paper, this is an Italian group, they found a huge infection in the liver, which was not so good, and AAV PHPB is only working in black cis-G mice. It doesn't work with the other strain of mice. And I have tested in the past AAV10, and it should work better, but in our hand, we don't know why, but the AAV10 vector didn't work. I don't know. Yes, there is also, could you try vector to improve BBB crossing? I mean, yes, we had previously a project to work with vectoris, but now I believe itself, this kind of technology, microbubbles and focus ultrasound, really, if we are really able to have 4-5 times increase, and you have to know also that we can, by using this technology, you can use this technology for AAV vectors, but also for any kind of molecule. I was thinking about BDNF that does not cross naturally the blood-brain barrier. You can open the blood-brain barrier also for this kind of molecule. And today, right now, currently, we are more trying to find the best, definitively the best parameters. And we are only pushing in this direction. Pharmacology, gene therapy, and opening the blood-brain barrier with these microbubbles. And we are also working, trying to use other bubbles. They are new bubbles that are even more efficient. And our colleagues, they provide us this kind of bubbles now. Geneviève,
there is a question from Bilal. Maybe you can ask your question, Bilal.
Do you hear me?
Yeah.
Hi,
nice to hear you.
Nice to hear you too. So it's quite impressive, the increase of number of cells containing this vector. But for MSCP2, the level of expression is very important also. So how you can manage the level of expression, because a lot of methylation is not good.
Yeah, you are totally right. I mean, this could be really like what I present. It's more like a dream. But to be honest, we are also looking, now we are just trying to have a maximum number of cells infected. But what we don't control, this is a number of copies of AAV vectors going in each cells. And we know that MSCP2 is a tricky protein, because if you don't have, you have in male, for example, you have epileptic, or you have early encephalopathic patients, they die pretty early. But if you have too much, it's not good. And this will be probably another problem that we will have to face. But the idea, we are working now currently with a codon optimized version of MSCP2. This will also lead to an increase of the expression. But we are thinking that we could probably use a codon disoptimized version to reduce the level of expression. That could be also something that we could use. But now we are just mainly working to increase and to have the maximum level of cells infected. And after we will see if it's too much, the level of expression, if it's too much.
Okay, thank you so much.
Thanks. I see, did you try to start the treatment at the pre-symptomatic period? This is a very nice question. I would say we know from a Scottish group, the same, they treated the mice just at birth, because of course this is a genetic disease and you can make a genotype at birth, and they injected the mice after I think this is the 3rd day after birth, and they succeed, they manage to have around 60%, 70% of cells infected, and it works perfectly, and the mice are totally rescued. This is good, of course, in this case, this is a proof of concept. But to treat before the appearance of the pathology, in our case, this is not what we selected. We I mean, we know that it will be harder, but our idea is just to treat when the symptoms start to be exactly in the same condition than for patients. And this is our strategy. Yes, and also, yeah. Yes, and yeah, Laurent, yeah. This pathology also, this is, we have to treat the girls, the red girls, because the pathology, this is de novo mutation, and there is no familial story. And this is why it's difficult to anticipate that, that parents could have a baby with a Rett syndrome. And this is why we are really pushing hard just to treat mice just after the beginning of the pathology. Yes, Geneviève Rougon, superb work, thank you very much. If I understand correctly, in female, half of cells express MCP2. Yes, this is true. If you deliver MCP2 to all cells, gene sur the, yeah. The problem, yes, in females, since this is X chromosome, MCP2 is located on the X chromosome, half of cells. This is why the females are living longer, and even if they have a red phenotype than the boys. And this also will be a problem. There are some strategy ongoing, but complicated. But the idea, it will be complicated to decide. I mean, to decide in what kind of cells we want to infect. Right now, currently, we are working like blind. We are trying to infect the maximum of cells, and we know that if a cell is already expressing MSCP2, it will be an overdosage. That could be a problem. But we know from also this many studies with mouse models that the level of MSCP2 has to be between 0.3, to be more or less correct, to 1.5 around this level. And we are also thinking about to modify the promoter that we are using, even to have a very low level, promoter with a very low level of expression, to don't overdose age and to don't go from a syndrome of Rett syndrome to another syndrome. This is a duplication syndrome. This is not our goal, but we are working in this way. But this also is a problem. And could you couple fuse with imaging? Yes. In our laboratory, we don't have MRI, but they are in USA. There is one group, one big group. They have focus ultrasounds and MRI. And they are working and they are able really to precisely select exactly the area of the brain that they want. In our case, one colleague in the laboratory of mechanical acoustic is working pretty hard to, because the bubble that we are using, this is the one that people are using for echography. And we could use this way to, at the same time, to see through the skull the different arteries and veins, and then to, if we know where they are located in the brain with a sort of map, we could also, by this way, select a special area in the brain. This is another solution, but MRI, of course, we don't have, but it's possible to couple. There is, yes, did injection in female have any effect on weight? We haven't seen we haven't seen it changing in females. The females are quite variable. We have very pretty big females, really big females. Some of them are normal, just normal. But we haven't seen. We checked, but we haven't seen any effect on the body weight. Clément Menuet, thanks, Jean-Christophe. Can you permeabilize temporarily the BBB in the brainstem as well as in the other part of the brain? I know why you are asking this question about breathing dysfunctions. And this is what we are looking at the beginning. We only focus in the central part of the brain, like I show the counting. But now we are really looking at the brainstem level and also cerebellum for some other reason. And it seems that the fuse, even if the focalization, it's quite complex. It's more because it's a little bit tricky. The skull has a shape, a special shape, and it's pretty complicated to target the brainstem. But we are working in this direction, and our very last results, the results that Marie-Solène obtained a few days ago, seems to show an increase of the level of infection in the brainstem, probably less than in the middle of the brain, but yes, an increase. It seems that there is an increase, but we have to count now carefully and to see how much we can gain, how much we can increase the level of infection in this area. Do you see a rescue of the respiratory alteration that is somehow proportional to the degree of MSCP2 expression in the brainstem? This is really tricky because the problem, even in the brainstem, if you look at the level of expression, I published a paper, it was 10 years ago, even more, just to look at the level of expression of MSCP2 in the different areas of the brainstem. And if you look, the level is huge, is very variable, and it can go from 1 to 10 times the level of expression, depending if you are in the locus coeruleus, if you are in the ventral area, if you are close to the nucleus tractus solitarius. And the level of expression is complicated even in the wild type mice. And the second difficulty, this is because, like I told before, when you use AAV vector, and especially with the focus ultrasounds, you don't control how many copies of the virus will reach the cells and will be expressed in the cells. Probably in some cells, you will have 2, 3, 4 copies. In the neighbor, it will be totally different. And this is one big problem, one big issue that all people using this technology and also the gene therapy has to face. This is to determine cell by cell, the level of expression. I believe it will be really tricky or almost impossible to do it.
We just have a last one from Lydia Karkarian.
Yeah.
In normal brain, is MECP2 homogeneously expressed in brain regions, cell population?
No, depending if you look in the brain, like I told for the brainstem, but if you look in the hippocampus is a little bit tricky because the cells are so small that you feel that the level of expression is really dense, is really important. But if you look almost everywhere, striatum, cortex, you can see that there are some subsets with very high expression, very low expression. And the idea that we had before in our previous paper was to try to correlate a high level of expression with a possible, in the knockout mice, a possible defect in the function controlled by such area. And we haven't been really successful in this direction, but yes, the level of expression is definitively different depending where you are looking in the brain. Yeah.
Okay, you have a last one from Miriam.
How do you explain full recovery even when tamoxifen is injected at very late stage of the disease? It means MCP2 is tricky, but it seems that the protein is not necessary. I mean, this is more or less like a dogma. MCP2 is not necessary in the embryo. Just during several weeks after birth in rodents, there is also no need to have MCP2. And MCP2 is more able to maintain an adult activity. This is the idea. Nobody knows really what MCP2 is doing, even if there are many, many laboratories. And to be really honest, the story with tamoxifen, I presented this fully recovery. It's really like a miracle, but we know, I mean, this has not been published, but this is something that we got, you know, in Congress, that before, when the mouse are sick, there is a part of leakage, and it's not fully a knockout mice, but the difference between before tamoxifen and after tamoxifen, it's really like a miracle. I think if you have time, it's very nice to look at the paper, because even the connectivity of neurons are rescued, the electrical activity is rescued, the body weight, so many different parameters. There is only one parameter that is not rescued. This is the skull, because the skull, there is a sort of microcephalia when MCP2 is mutated. And when you reactivate MCP2, it's difficult to explain how the brain could push the skull to have something increased, to have a skull increased. And this is only one parameter, but almost all in vivo, in vitro, by electrophysiology and by biochemistry in this fantastic paper published in Science. It looks like really like a miracle, and the only explanation, MCP2 is only important in adults to maintain the shape and the function of neurons yeah. But we have no more information.
Thanks a lot. Thanks a lot, Christophe. So I'm just gonna share my screen.
Thank you so much. You're welcome. I don't know how, yeah.
So thanks everyone. It was really a good session with a lot of questions, and it's very, very interesting. So we will meet you in June. There is no Neurobinar in May because there is NeuroFrance. So we encourage you all to go to NeuroFrance. It's a virtual session, meeting, sorry, but it promised to be very interesting. So we will meet you on June 17th at 4 p.m. to meet the Phenotype Institute, specifically the Mosaic team, which is integrated with the Neuromarseille Institute. It's really new, so it's a good occasion to meet other colleagues. So thank you all, and meet you on June. Bye-bye.
Okay, bye-bye.
Thank you, bye.
Bye bye.
Bye.